Welcome students. My job today is to explain to you how lipid membranes are directional. So one of the misconceptions that takes place when reading the textbook is the orientation of the lipid membrane when it comes to the side facing the cytoplasm and the side facing the non-cytoplasmic surface. So let's draw that. So let's start with a cell. So here we have a typical cell, and this cell is surrounded by a membrane. Now assume that we're looking at a eukaryotic cell, and as we know, eukaryotic cells have internal membranes as well. So the nucleus has a double membrane. The mitochondria, they also have a double membrane, and some other organelles too, like chloroplasts. But most internal structures that are surrounded by membranes have a single lipid bilayer. So let's have a look at the structure of this particular membrane. Whether it comes from here or here, it's basically the same. So let's draw that. So we have these phospholipid molecules which are orientated in a particular direction when it comes to the structure of the membrane. So we have a lipid bilayer as indicated in this diagram here. I'll just finish this off. So here's your lipid bilayer. It contains two leaflets. The top leaflet, let's call that the cytosolic, and the bottom leaflet here, and let's call that the non-cytosolic. So the goal of today's lesson is to explain to you how these two surfaces maintain their directionality as membranes are shuffled back and forth between different regions of the same cell. So where do we start? Let's begin by redrawing the structure in a much larger version. So we'll get rid of this. And then at the end of the video, we'll talk about what happens in prokaryotic cells to compare them to eukaryotic cells. So I'll use a different color here. So to pretend that we have an internal structure like a liposome. So here's your liposome. And this liposome could be any structure. It could be a lysosome, it could be a peroxisome, and it could be any kind of vacuole inside the cell. So this one single line here represents, when magnified, these two leaflets that make up the surfaces. So if this is the cytoplasm or the cytosol of the cell, and this is the lumen of this structure here, this organelle, then we can orientate these molecules within that membrane by giving them a label. So this side here, the top surface, will be facing the cytosol. So here we would have the cytosol, just like we do facing the top surface of that membrane. The non-cytosolic surface of this leaflet, which is also called the lumen in this particular case, will be facing this side. So once again, just put this in context, this lipid bilayer is actually this thin line represented in this diagram here. So the cytosolic face may have different proteins and different lipid molecules in its composition. So maybe these ones here are different to the ones below, which will be a different species of phospholipid. Let's just color them in. If you can see the colors, that's great. If not, we'll have to assume that the two species are different populations. I'll just give you some words to apply to this from the textbook. It could be that here we have more lipids, phospholipids of the serine type, phosphatidylserine. Whereas here we could have more of the choline type, phosphatidylcholine. And we'll understand later in this video how this membrane can be asymmetrically constructed inside a living cell. But for now, let's just concentrate on the surfaces and how those surfaces maintain their integrity. So what I need to draw next is the membrane moving away from this location to fuse with the surface of the cell. So let's draw the surface of the cell here. So the surface of the cell is indicated by this line here. So once again, this is going to be your plasma membrane. And the goal is to move the membrane from this structure and have it join this structure by transversing the cytosol. So we're going to use two colors by just amplifying this to mimic what's going on here. So those two colors, if they come out in the video, will then indicate the orientation of that membrane. So let's just get rid of some of these words and labels. So what I'm doing now is I'm redrawing that structure, but with two lines to represent each one of these leaflets. So we'll have the inner line be the non-cytosolic surface of the lipid bilayer. And then if I use a different color, I'm hoping that the alternative color will show up, and that'll be the cytosolic lipid leaflet on the outside. So we have a single organelle surrounded by a lipid bilayer indicated by the two lines. Now as we migrate our lipid bilayer away from here by pinching it off, and we'll indicate that by redrawing these lines in this fashion. There's one, and here's the other. We notice that the interior leaflet, this one here, once it's pinched off, is still the interior leaflet. The exterior leaflet, this one here, it's still the exterior leaflet of this new vesicle. And as the vesicle moves, it will maintain that orientation. So let's redraw that. So here's the vesicle a few minutes later as it migrates through the cytosol. So the two leaflets are maintaining their orientation. And as this vesicle fuses with the plasma membrane, which again is also made up of two layers of lipid molecules, and let's redraw that again to match what we've done over here so that we maintain consistency throughout our diagram. So the plasma membrane will be redrawn with one line, this one here, to represent one leaflet, and we'll use the second line to represent the second leaflet. The one on the inside is again the cytosolic facing surface, and the one on the outside is, in this case, the non-cytosolic facing surface. In this case, it faces the extracellular environment around the cell. So let's follow the migration of this particular vesicle as it begins to fuse with the plasma membrane. So let's redraw that a bit further along in its path, maybe here, and here. So this vesicle has now migrated from here to here and is now going to add more membrane so the cell can grow in dimensions. So we're growing the cell by bringing in more membrane from the interior of the cell. So what's going on here? Well, in a very sophisticated maneuver, the membrane that makes up this vesicle is now going to join the membrane that makes up the plasma surface. And these two will somehow, in a very complicated fashion, blend together, as indicated here, like that. And you can see what's going to go on. As this vesicle adds its membrane to the surface, the membrane is going to expand by that much. But here you can see the inner non-cytosolic surface of the lipid bilayer maintains its integrity. It's on the inside, it's on the inside, and then it appears on the outside. That in itself is important, but if we follow this one, it makes much more sense. So here we have the outer leaflet, the cytosolic facing surface. When it forms the vesicle, it's still on the outer surface, facing the cytosol. Here it maintains that cytosolic facing direction, and when it blends with the membrane, it will also be facing the cytosol. So if we had a certain species of lipids in here, that were facing the cytosol, they would maintain that orientation. If we were to organize this new membrane here to have a completely different combination of phospholipids, as you can see, the lumen and then the extracellular surface would be maintained. So if we now populate this structure with some proteins, and let's draw the proteins diagrammatically to represent a couple of different shapes. So the triangular proteins are only found facing the cytosolic surface, and let's assume that the circular proteins are only found in the non-cytosolic surface of that membrane. We can now translate this to this structure here. So as we said here, these proteins, the circular ones, would only be found attached to the inner leaflet of that membrane, whereas the triangular ones would be found attached to the exterior cytosolic facing leaflet of that membrane. So if you want to take a few seconds, you should be able to pause the video and then label this, this, and this, and finally label this with the orientation of those two different types of proteins. So I've assumed that you took a few minutes to do your revision and figure out what happens. So let's see if you got it right. So let's follow these proteins as they migrate through the cytosol of the cell. So let's go with the orange. So the orange here would stay on the outer leaflet of the vesicle, and they would continue to be on the outer leaflet of this vesicle here, and likewise here. But now as they begin to fuse, they will now only be found on the inner leaflet of the plasma membrane facing the cytosol. If you go back to this scenario here, those triangular proteins should always be found facing the cytosol and never in the other direction. If we now follow the circular proteins as they are pinched off, they will only be found attached to the inner leaflet, and as the vesicle is pinched off, they will only be found on the inner leaflet, and when the vesicle starts merging with the plasma membrane, they will only be found initially on the inner leaflet, but as the merge takes place, they will be found then subsequently only on the exterior extracellular surface of the plasma membrane. So this is a very important concept that students must remember. As membrane is made, it maintains its orientation. Thank you so much.